Hello and welcome students. I am Anirban. In this session we are going to learn how to design small RNAs, mainly siRNA and shRNA, and how they regulate eukaryotic gene expression and promote gene knockdown. So first of all, we need to understand what is a gene knockdown. Gene knockdown is an experimental technique by which gene expression of an organism is reduced or deactivated. So that means it is a suppression of a particular gene rather than deletion. Now, gene silencing, gene editing, and conditional gene knockout are forms of gene knockdown, and it can be complete or partial. And gene knockdown is mainly done for studying gene role in the biology of the organism. Now, there are so many techniques that are used for gene knockdown. Among them, RNA interference, CRISPR, and tolerance are the most frequently used. RNA interference, it's basically a post-transcriptional modification which is triggered by providing a double-stranded RNA. Two types of RNA, small RNAs are used, that is microRNA and siRNA, and they play a central role in RNAi-based gene silencing. So actually, they are they mainly used homology-dependent control for gene activity. Next, CRISPR. CRISPR-associated genes encode cellular machinery uh, that cuts exogenous DNA into small fragments. On the other hand, tolens are the small nucleases that have two important functional components, a DNA binding domain and a DNA cleaving domain, and they can be designed specifically to cleave specific site. So in this session, we are mainly focusing on RNA interference. It's a natural method of gene regulation, and it is a promising tool for studying gene expression and to treat various diseases like viral infection, cardiovascular disease, cancer, and metabolic disorders. As already I have mentioned that it's a post transcriptional gene silencing process where double-stranded RNA is introduced into the cells, and by using the natural machinery, it dictates sequence-specific gene. Then RNA silencing phenomenon was first seen in C. elegans, where per1 mRNA was degraded, followed by introducing double-stranded RNA into the nematode worm. Uh, RNA silencing, it's very specific, and very small amount of double-stranded RNA is sufficient for silencing gene expression. Previously, I have mentioned that miRNA, shRNA, and siRNA plays a central role in RNAi-based gene silencing. Here, our main focus is mainly on siRNA and shRNA, how they regulate specific gene expressions. Now, we are going to study the mechanism of action for siRNA. Endogenous enzyme dicer first cleaves a long double-stranded RNA into smaller fragments and forms siRNA. This siRNA then associate with an RNase containing complex to form the RISC. RISC is RNA-induced silencing complex. Then RISC unwinds the duplex and releases the sense strand. The risk bound antisense strand then serves as a guide for targeting the activated complex to complementary mRNA sequences. This results in subsequent mRNA degradation. For shRNA, when it is delivered to the cells, it gets integrated into the genome, and the knockdown effect is passed onto daughter cells. The mechanism of action is similar to siRNA. First of all, shRNA is cleaved by dicer enzyme to generate the desired siRNA duplexes, which in turn associate with RISC complex, and the rest of the path is same as siRNA.
Sorry, sir, the voice is.
Yeah, actually, this particular slide has no voice actually. It is another way of representing the shRNA mechanism. So shRNAs can be transduced to cells with lentiviral or lentiviral vectors basically. So after transduction, unlike siRNA which goes to the cytoplasm and does it function, these transduced shRNAs are located to the nucleus where it forms pre-shRNA, then it is processed by DROSHA, which converts it to shRNA. Through exportin 5, it is exported to the cytoplasm, which is then processed by dicer to form siRNA. So ultimately, the mechanism of action is through siRNA only. This siRNA is then processed by the RISC complex, RNA-induced silencing complex, where there are guide and passenger strand. The antisense strand is left and the other strand is destroyed, and this particular guide strand, it takes it to its respective mRNA. It goes and binds there. Then there are different RNAs present in here, which will ultimately degrade your mRNA. So through siRNA, you directly transfect it. Through shRNA, you can transfect it through plasmid-based vectors, as well as you can do transduction as well. Then this is a simple miRNA biogenesis mechanism. Not much to look at. Like miRNA, from the gene, it firstly forms primary miRNA. miRNA is not your of your interest. You are more interested in like designing siRNA and shRNA for a gene of target. So basically, since it comes under the wide umbrella of non-coding RNAs or RNAi mechanisms, that is why a simple slide is given. Primary miRNA is converted, processed to precursor miRNA, which is exported to the cytoplasm. This precursor is like cleaved through dicer similarly, and RISC takes it to its respective targets and degrades it. So here is a short video that is will be helpful for your understanding of this mechanism of action of RNAi. I'll just play it. Look at it very carefully.
This animation explains the unique Benetech ddRNAi technology and how it is different from siRNA, the more commonly used RNAi approach. In a normal cell, genes composed of DNA in the cell's nucleus continuously produce RNA, which codes for specific proteins. In disease, sometimes there are rogue genes which are turned on inside cells, and they make mRNA that produces proteins that shouldn't be there. These proteins cause the cells to malfunction. One approach to treating the diseased cells is to turn off or silence the disease-causing gene by targeting its mRNA. This is generally done using short interfering RNA, or siRNA. siRNA uses synthetic double-stranded RNA molecules which enter the cells. An enzymatic complex, or RISC, then finds the siRNA and separates it into single strands. One of the strands, the guide strand, remains with the complex. The other strand, the passenger strand, degrades. The guide strand then binds to the target mRNA, leaving it at a particular site. The mRNA then degrades. This method is effective, but the siRNA must be re-administered regularly to maintain the silencing of the disease-causing gene. Benetech has developed a technology that can provide permanent silencing of the target gene with a single treatment. The Benetech ddRNAi technology is a DNA construct that is delivered to the cell using a vector. The DNA construct enters the nucleus of the target cell. Double-stranded RNA in the form of a short hairpin, or shRNA, is then continuously expressed from the construct. The shRNA passes through the nuclear membrane into the cytoplasm of the cell, where an enzyme, DICER, chops off the loop. This converts the shRNA molecule into an siRNA molecule. The process from shRNA production to mRNA cleavage continues indefinitely for the life of the cell. This ensures that the mRNA can never be active. Since the ddRNAi process results in the cell continuously expressing the shRNA, there is no need for further dosing, effectively providing a cure to the patient from a single treatment. This animation
Here, a brief
Sir,
point. Yeah.
sir, this shRNA is responsible for turning of the disease-causing gene, turning of the gene which is responsible for the disease.
So the main difference between siRNA and shRNA is siRNA is transient while shRNA will be stable. What do I mean by that? Basically, siRNA and shRNA both will knock down your gene or shut down your gene. Not completely. देखो, complete deletion is called knockout, which we are not doing here, which is usually done by CRISPR. But this siRNA or shRNA, these are used for knocking down, which means it will decrease the expression of a particular gene to a certain extent, maybe 80%, even 90%, but it cannot completely knock out a gene. Okay, so for that you would need different machinery. Here siRNA will disappear after few cell division. Like if you transfect normal siRNA to cells, it will disappear after few transfection cycles. Sorry, after few cell division cycles. But if you put shRNA, it can be stably integrated into the chromosome, which will be in each as long as the cell will live, it will be continuously making this particular shRNA, which will be ultimately converted to an siRNA. And whenever that particular gene will be transcribed, it will find its miRNA and degrade it. So basically, it is a stable source of like degradation of a particular transcript. So that is how shRNA is better. But shRNA designing, not designing, its delivery is a little bit complicated, and the vector choice of vector you need to be very careful about it. So in this particular slide, there are a few differences between siRNA and shRNA which you need to understand well. Like just go through it, not much detail to say, but still I'll like just read it out to you. If you have any doubt, just you can ask me. Single stranded and so siRNA is a single strand. Ultimately the final form will be a single stranded RNA molecule produced by the cleavage and processing of the double stranded RNA. Here it is a short sequence of RNA which makes a tight hairpin turn and can be used to silence gene expression. Those stands for small full, just the full form, small interfering RNA, short hairpin RNA, produced in the cell naturally. So siRNAs are produced. Oh, sorry, trying to get the pointer, but apparently it changes. So it is produced in the cell naturally. So siRNA, miRNA, all these are produced in the cell naturally. This is produced in the cell artificially when you are introducing the corresponding RNA genes through transformation or transduction, even transfection. A double strand. So this is a double-stranded RNA molecule, usually 21 to 25 nucleotide long. It is like, since it is a hairpin, it falls back on itself, and it is about 80 base pair in length and includes a region of internal hybridization which creates a hairpin structure. This is short, this is comparatively longer. This forms a double-stranded RNA with a 3 prime. So this part is important, with a 3 prime end overhangs of two nucleotides, which activates the RNA interference. And this particular shRNA contains a loop structure which is processed to siRNA. Ultimately, it will also be processed to siRNA. Okay. This particular siRNA is produced by the processing of miRNA either by dicer or DROSA proteins endogenously. This mimics miRNA. So just ignore this particular point, does not make that much sense. This is transient, like what I was talking about earlier, by means of the effect per dose, and this is more sustainable. Like if you give it to the cells one time, it can be there for longer period of times. siRNAs are effective at high dose. These are effective at a low dose, has a low potency, has a high potency, has a simple manufacturing process, results in less off-target effects embedded in a miRNA scaffold. So, but this particular point means that your siRNA, since it will be in abundance in the cytoplasm, it might bind to some other genes other than your gene of interest. So in that sense, it is a little less specific. So you should take good care while designing siRNA, so that it becomes specific to your gene and it randomly doesn't go and bind to other healthy or good genes in your body and knock them down. So that is not intended. Can be used to short-term knockdown of proteins by the direct application of the target, can be used to long-term, the same thing over and over again. So that was the difference between siRNA and shRNA. Now I will move on to si. Okay, I need to.
So previously we have seen how siRNA and shRNA are processed in the cell and how they regulate target gene expression. Now to target any specific gene, we need to design specific siRNA, and for that we follow some guidelines. First, choosing the siRNA target site. It is the most important part. Regions about 50 to 100 nucleotide downstream of the start codon AUG of the target genes are preferred target. Then target sites starting with sequences like 5' AAN 19TT, 5' AAN 21, or 5' NAN 21 on the target sequence are preferred. Then duplexes of 21 nucleotide RNA with 2 nucleotide 3' overhangs are generally used in RNAi experiments. But 19 to 29 nucleotide length is okay. Then it is better to avoid single nucleotide polymorphism, intronic part, and 5' UTR and 3' UTR as a target site. GC content should be less than 50%. It should be 30 to 50%. If the target sites are forming some secondary structures like internal repeats, palindromes, then it should be avoided. The secondary structures can be predicted by software such as mfold. So it is already mentioned that overhangs are very necessary for siRNA designing, but one should avoid G residues in the overhang. Next, sense and antisense strands should have the ability to form the duplex properly, and at least 4 out of the 7 nucleotides in the 5' end of antisense strand should be A or U. The repeats like GGGG or CCC should be avoided to decrease the risk of forming hairpin structures. After designing siRNA, the sense and anti-sense both the strands should be checked via blast with the RefSec RNA database to avoid to reduce the risk of silencing unintended genes. Then, as their alignment may not be the result of chance, blast results with smaller e values should not be overlooked. Then less than 80% query coverage with other genes less than equal to 15 nucleotides out of 19 matching with the respective siRNAs is believed to be tolerable. Then siRNAs are synthesized, then transfection uh, lipo and for transfection lipo and peptide based methods are used. Then detection, once it is delivered into the cells, the quantification of mRNA and protein of the target at the mRNA and protein level of the target gene is very necessary uh, to analyze whether actually the siRNA is silencing that particular gene or not. After designing these are some online software available for designing siRNAs. For siRNA designing, we are going to use SI Direct, this software. It's an online tool for designing mammalian siRNA using the algorithm by Wei et al. The most important parameter of SI Direct is its capability to create and use a non-redundant sequence set of genes in order to decrease the effect of alternative splicing. By this non-redundant database, users can design a siRNA that is specific for just one transcript variant of the desired gene. Next, this database is qualified for a form of Unigene and RefSec databases, and it includes sequences of duplicate-free exons and exon-exon junctions. To avoid to reduce the off-target effects, the temperature is set less than 21.5 degrees centigrade. That is, TM should be less than 21.5 degrees centigrade. U-tie rule, there are four rules, that is A and A or U at position 1 from 5' terminus of the siRNA guide strand. Then second point is G or C at position 19. C and third point is AU richness in position 1 and 7. And 4 is no longer DC stretch less greater than equal to 10. So is direct follows these rules and provide some option of siRNA.
From here on, you follow very carefully, because I think your assignment also talks about designing siRNA and shRNA for your gene of interest. So I'll be playing this. Follow this part very carefully.
For siRNA design using SI Direct version 2.0, first of all we need to search of our gene of interest and NCPI. Here we taking MYC as our gene of interest. Then if you open that, you will get a page like this, and here you can get information about your gene of interest, also known as summary function, etc. Now, if you scroll down the same page, you will get available isoforms of your gene of interest. For example, here for MYC we got two isoforms. uh, two isoforms, and we are going to design siRNA against isoform 2. So we need to copy this highlighted code that represents the mRNA sequence of MIC. Then you just need to paste that the mRNA code in SI Direct software, and to change any parameter, you have to go to option, and then according to you can change some parameters, and then click on design siRNA.
Here, when you go to this particular page, you can hover your mouse over these question marks. This will tell you the purpose of this particular step. Okay, so if you have any questions in mind, you just put it here.
Now, if you click on the design siRNA, you will get a table like this that contains so many options of siRNAs. From this, you can choose your desired siRNA that targets a MYC isoform 2. You can use that guidelines to choose your siRNA that I have mentioned earlier, and you also can get some more informations like target sequences, TM, etc. from this, and uh, and the target position from this table.
So when you do your assignment, you give like at least two siRNAs for one gene, and mention the properties of the guide strand and passenger strand. Like from here, you can get the TM of those strands, and the position, target position, and the target sequence also. So do all these things. You need to make sure you put it in your assignment.
So previously we have seen that how siRNA are designed, should be designed. Now shRNA designing. First point is choosing the shRNA target site and specificity checking. It is same as siRNA. Then synthesis of that specific shRNA and packaging of shRNA, encoding lentivirus. So to delivering this shRNA into the cells, we require some plasmids, plasmid specific for shRNA delivery. We need to clone that the oligos in the specific plasmid, and then only can we can deliver that shRNA into the cells. So, for example, there are so many plasmids are there. We are taking PLK1 Puro as an example. So PLK1 is a replication in incompetent lentivirus vector delivering shRNA. PLK is can be introduced into the cells via direct transfection or can be converted into lentiviral particles for subsequent infection of a target cell line. Once introduced into introduce the purine resistance marker encoded in PLK1 allows for convenient stable selection. So the difference between siRNA and shRNA is that siRNA is transient, whereas shRNA forms stable silencing of that gene. Then PLK1 contains 1.9 kb stuffer that is released when digested with EcoR1 and AGE1. That is the restriction enzymes, and we need to make oligos that with the overhangs that can bind with this the sticky ends so that they can clone in so can they can like it.
So here you need to understand that basically this particular sequence, it is directly like your sense sequence, right? So it will match with your the target site that you want to like design siRNA or shRNA for. Then there is a loop which forms this particular part of your shRNA hairpin. Then this particular thing will fall back on this red, this green. Okay, this green will fall back on this red. So basically, it is complementary part. So there are repeats of it, and then basically that will be your forward oligo, and then you take its reverse complement, which will be your reverse oligo. Then it and there will be two overhangs like double T or W in case of RNA, which is required for the dicer binding and all. So maybe in next slide you'll get a better idea. Let me take you there. Okay, I'll just play first, then I'll discuss.
So designing oligos, shRNA oligos are the most important. To generate oligos for cloning into PLK1, you need to insert your sense and antisense sequences. You shouldn't change any ends. As these bases are important for cloning the oligos into the PLK1. And now if we take MIC as our target gene and the sequence written in red as target sequence, the oligo should be like this. And once it is transfected in the cells, like siRNA, the siRNA for shRNA also the level of target gene should be analyzed, so that to analyze should be analyzed to see whether the shRNA is actually silencing that particular gene or not.
Okay, guys, so do not get confused. Okay, so this is for MYC the region that you want to. This is basically what is called the seed region. So first you will put this CCGG. I don't know if you got it, but this is the restriction enzyme sequence. Okay, so it is this particular AGE1 restriction enzyme CCGG. So you can choose any enzyme which any two enzymes which are actually compatible with each other. So in this particular scenario, they have chosen AGE1 in the forward prime oligo and EcoR1 in the reverse oligo. So as you can see that the rate sequence remains exactly the same. So listen to me very carefully. This part you need to follow for designing your own shRNAs as well. So siRNA you will get from the software itself. shRNA you do it like that. So you put your enzyme site, restriction site, then your sense strand, then this particular thing will form the loop. You can take it from this sequence also, and then if you look carefully, so it falls back. It is a reverse complement of this particular sequence. So here C is converted to G, then G is complementary to C, then there are 1 2 3 4 5 6 A's, and you will find here 6 T's, followed by there should be double T, so here it will be double A. So as you got it, I hope this is a reverse complement. So basically this particular green sequence will fold back on this red sequence, and it will form a hairpin. This will form the neck of the hairpin. Similarly, the reverse oligo will be completely like, first of all you will have to put this restriction site double A double TC. So where is it? Yeah, double A double TC. This is the restriction site. Then you will put then the rest will be the reverse complement of this. So here there are like 1 2 3 4 5 6 7 T's. I think here should be 1 2 3 4 5 double A and then double G. Yeah, double C will is converted to double G, and then T A then G double T, yeah, like that. And then your uh, basically the reverse complement of the sequence. So the enzyme site goes in the 5 prime end in both forward oligo and revert oligo cases. So it's like that. Transfection will be same as siRNA, and detection you can do real-time PCR. It is same similar as siRNA. So moving on, let me see if there are any more slides.
So coming to the conclusions, RNAi is a natural method of gene regulation. This technique is it's a very promising tool for studying gene expression. Both siRNA and shRNA are target specific, and choosing target sequence at the time of siRNA and shRNA designing is the most important part. Though the mechanism of action is similar, siRNA is transient, whereas shRNA is stable.
So one very important take-home note is when you are designing siRNA, I hope most of you will use SI Direct. But if you use some other tool, read about it, understand it completely, then use their algorithm. Okay, if you are using SI Direct, you are getting your forward and reverse like the both strands. You need to do a blast like NCBI primer blast. You go there and do a blast to see whether it binds to some other gene. Now it might bind to some other gene, then you have to look for this complementarity. If more than 15 bases are complementary to that other gene, so this your siRNA might be non-specific. So if you submit, if you just try to be smart and put the forward and reverse sequences, and your evaluator puts them onto blast and sees that this particular pair of siRNA, I mean forward and reverse siRNA primers, I mean oligos, are binding to some other gene, then your marks might be deducted. So be smart about it. Go to NCBI primer blast, put the two sequences, and look, just click on get primers. Okay, NCBI primer blast, you can go put the forward and reverse sequence and get primers. Then you will they'll return you some possible binding where it can bind with your gene. So if it is binding to the gene of interest, what you are predicting, it will definitely bind to that, of course. Then if it is binding to some other gene, then you look for the amount of, I mean, how many base pairs it is binding. If it is binding perfectly complementarily with more than 15 or 16 bases, then non-specific effects might come. So take care of that. Other than, there is not much to do. You just look for the TMs, the conditions, if they are fulfilling. Try your best to design as close as possible. And shRNA, I already told you how to design it. So that's it, not very difficult. You will also get your transcript sequence from, you have already discovered that, I hope, through NCBI or FSEC. So it's easy. Now you guys can ask questions. Okay, no questions. Am I still connected? At least tell me that, guys.
Yes, you're connected.
Okay, thank you, thank you.
Yes.
I hope it's clear, right? No, I don't think any I mean, problem should be there designing it. It's quite straightforward. Anybody? Any doubt? Okay, maybe then I'll stop the meeting. Okay, guys. Then I'll close it. I hope, wish you guys all the best, and I hope you don't face any problems regarding this, but these two assignments. It should be easy, and all the best to you. And I'll close it. Anybody? Just give confirmation.
Okay, sir. Thank you, sir.
Thank you,
Thank
Anil.
you, sir.
Okay, bye
Thank
bye.
you, sir.
Bye bye.
Sir.